All right, hello everybody. I'm Brian. This is AAMI News and Conversation. We're here at day 3 of Kilmer 2025 in Orlando, Florida. And I'm here with some great guests. You probably recognize Ralph Basile of the AAMI Foundation. And we're also here with William Leiva, which is, I'm excited about this one because I remember thinking about your research in 2022. You had received a foundation grant. So Ralph, what was that grant for, and what are the grants all about anyway?
Sure, and I'll ask Dr. Leiva to go into detail. But as you know, we have a couple. The AAMI Foundation has a couple of research grant programs, and one of them is the Kilmer grant. When of course we're here, as you said, at the Kilmer conference, and that was started with donation by J&J to start the Kilmer Fund. And it's meant to be seed money for research that's going to help advance the industry and maybe be a start of further research. So, Dr. Leiva, maybe you could explain your 2022 grant.
Yeah, absolutely. So first, it's a great opportunity. I was doing my research as part of my PhD program, so I needed to publish, as everyone knows, right? So you need to publish during the PhD. And one of the driver was to explore the what if medical devices were to be manufactured with materials that eventually have antimicrobial properties. That was the whole rationale. We did that in collaboration with Dr. Zuniga from University of Nebraska and Dr. Laranjeira as well. Both of them have a really good background in terms of research, but also understanding medical devices and 3D printing with these really novel polymers. So it was very interesting to explore how that was going to behave after sterilization and see the performance of the materials as well. So that's how everything started from that research, initially driven because I needed to publish, but then it kicked in immediately my interest in the reprocessing world and definitely sterilization of medical devices as well.
Okay. And the purpose of that, and it's a coating, is that correct?
It's a copper infused polymer.
Okay.
So you can use it in many ways.
Okay.
We use 3D printing because it's cheaper to manufacture
Yeah.
rather than using injection molding, but you can use it like any thermoplastic.
Okay. Because copper has antimicrobial properties,
It sure
so
does.
it could be a good either coating or substrate for a medical device or other products. Is that right?
Absolutely, and
Yeah.
that was the initial thought. When we think about the challenges we face in the industry, think about flexible endoscope, for instance, right? If we were to have something that were to decrease the microbial activity of the bioburden that we get out of patients or because of patient procedures, it would be fantastic. So that was the initial thought process and potential applications, but we needed to build the evidence and whether this performs in the right way. There's some discussions about the potential biocompatibility because copper is a metal and eventually may deliver some adverse event or toxicity, but that's further research. I mean, we need to continue expanding that because the material is there. It's not proprietary, so anyone can buy it. You can buy it on Amazon if you like, right? So you can make it easier to prototype very quickly and assess biocompatibility and toxicology.
Okay.
There's a precedent for this though too, like the use of copper. Like I immediately thought of the International Space Station where they're using copper for the microbial properties you mentioned. But did you take a different approach in developing this specific copper material, or is it a very similar approach that they do for aerospace?
It's very similar. It started with that thought process, right? So what are the needs of the International Space Station in terms of materials, right? They don't want to ship medical devices up there because it's very expensive. Just to send 1 kilogram or a couple of pounds to space is extremely expensive. So what they're doing is they're shipping the material in and printing whatever they need up there. So if you don't use it, you keep the material there, right? As opposed to if you don't use the medical device, you still have to ship everything and keep it there in stock, and space is limited too. So that was the initial discussion we had with Dr. Zuniga back then and tried to address that specific need.
Okay. Oh, that's fascinating. So they're using that technology on the space station now, or it's still
I don't know if they're continually use that, but they have used that in the past.
They have. Okay. Yeah. That's fascinating. Where do you we move on to the next your latest project, but where do you see this going then? I mean, what are you expecting?
It depends. I mean, when we think about the Kilmer community, we're exploring the challenges the industry faces, right? So one of the challenges are related to sterility assurance, of course, microbiological quality, but also materials. So if we were to think that this is a potential material, maybe not the current polylactic acid infused with copper nanoparticles, but maybe a different kind of polymer infused with nanoparticles of copper or maybe silver or some other active ingredient, right? Are we willing to continue exploring that avenue? Are we willing to explore that? Because it's a more expensive material for sure, right? But there's added benefits. So what if we are able to reduce the bioburden of materials and then reduce the amount of EO we have to put on the load for sterilizing that, thermally sterilize those devices? So I think there's a lot of what if, and I think it will need a lot of courage from us as industry to explore that.
Gotcha.
Where we want to take this technology that is available there. There's a lot of solid evidence supporting its use. Do we want to explore that? Or we want to continue exploring the pathway we have over the last many decades, right? Same materials, very similar polymers, similar sterilization processes.
Gotcha. Yeah, no, fascinating. So at some point, perhaps a company will come along, commercialize, or multiple companies. You said the material is readily available on Amazon. I think of the internal, I don't know if this is a good example, but the internal, you mentioned endoscopes, but PTFE tubing that, when it starts out, it's fine, it's intact, but as soon as it starts getting used, you start getting cracks in it and gouges and that kind of thing, and then that's where, And
we think about like lumens or suction devices, right? We think about them like very, very smooth surfaces, right? But at microscopic level, there's a significant roughness, and we were talking about topography of the surfaces a couple of days ago during this conference. And when you think about that, as long as you have surface roughness and if you have residues, bioburden and whatnot, something will happen. Right. And chances are, if you're talking about an endoscope, chances are it's going to be a biofilm, and that's not good, right? There's several consequences of that. We all know about it, but if we cannot manage the surface roughness, the integrity of the film in these lumens, very, very slim lumens, why not trying to address that from a different perspective? Why not try to reduce the microbial activity? Because we know that roughness is going to be a problem no matter what material we use. Injection molded, whatever manufacturing process is going to be, whatever polymer we use is going to be a challenge. So what if we provide these antimicrobial properties to that film or to that tubing that we need in medical devices?
Yeah. Great. Like I said, a new way of thinking about things. And as you said, it does make me think of the presentations we've seen during the Kilmer this week and trying to come up with innovative ways of dealing with cross-contamination, infection, making medical devices safer. And so what your research has done is just that, right?
And I think this is a very great opportunity for researchers, whether you guys are pursuing research for a PhD program, a master, or whatever. It's very interesting because industry is in general very conservative, right? We like to work with reliable technologies that are currently available, right? So this is a low risk kind of grant that allow us to think a little bit differently, right? So what if we try this new material? What if we assess the impact of superheated steam on sterile barrier systems and whatnot? So I definitely encourage anyone, and we see this on AAMI Exchange in different communities across AAMI and Kilmer community as well. There's a lot of folks really eager to make discoveries and apply science and continue the exploration and experimentation. So this is a great way to get funding that maybe our companies are not very keen to do because it's not within the line of work we're usually doing Monday to Friday, right? So this is a great opportunity. I definitely encourage anyone to apply.
That's great. So you just alluded, and maybe Brian, but if you don't mind, but so and one of the things I do let people know is just because you're awarded one grant in the past, you can apply for another. And Dr. Leiva, you did that, and maybe you can talk about your latest study that's in progress.
So, and just to bring everyone to context, I've been working for the last 15 or 16 years of my career in reprocessing, right, and sterilization, industrial and healthcare facilities. And I'm very aware of the challenges that folks face on the healthcare facilities, right? And all the tweaking and all the things that they do, they shouldn't be doing, but they still do, right? So that was, with that said, the rationale was, let's talk about superheated steam. We rarely hear about it. People don't think it happened, but it does occur in most of the countries, not only here in the US. So what effect that superheated steam may have on sterile barrier systems?
For clarity, when we talk about superheated steam, we're talking about it's steam sterilization within parameters, and then something goes wrong or process is changed.
Someone is modified, the process is altered in many ways, and the temperature is going to be a little bit higher, and that higher temperature drives towards a condition that is more closely related to dry heat. And everything in hospital, we talk about moist heat or autoclaves and cycles like that. So whenever you alter those parameters and we think about a sterile barrier system, they are designed and tested to withstand moist heat conditions, not superheated steam conditions. So that was the rationale of exploring what if superheated steam condition is altering the ability of the sterile barriers, particularly peel pouches, to maintain sterility over time. There's a lot of discussion around how long a package is going to be sterile. And depending on the regulation you have in different countries, people will say, well, if sterile barrier A is used, it's going to last 6 months. There's not a lot of evidence supporting that, but people do that. But if the sterile barrier system is damaged through the sterilization process or because of the sterilization conditions, would that affect the sterility of the devices, right? So that's the question.
Do you actually have to pull it out, pull that item out of service and they re-sterilize it because it was exposed to a condition? And what your research is looking into, well, do we need to do that? Because obviously there's a lot of waste. We're very focused on sustainability. When now all of a sudden you're using all the energy, time, and I mean literally energy and time and materials to once again sterilize something that may still be sterile, may be safe to use. Is that?
Or may not, and you touch in a very interesting point. So you mentioned, environmental impact, and we have been talking in the industry around sustainability for years, right? When you think about the conditions we have in the sterilization, conventional cycle for moist heat or autoclave sterilization, we're using a tremendous amount of water. Not for steam generation, but just for the vacuum that we need to create for conditioning the load and then for removing the residual condensate, right? So whenever we have to reprocess something, because something went wrong, a BI was positive, the CI didn't change and whatnot, we need to think about what the environmental footprint is going to look like. So over the course of this conference, we have been looking into the carbon footprint, which is very relevant and very significant for an industry like healthcare and medical devices. But we're not talking a lot about water usage, and that is a problem, and it will be a problem.
Sure.
Not in most of the U.S., but if you think about some other areas where the certification is expanding, access to water and a decent quality according to what we need for reprocessing purposes is limited. And it's going to be more expensive. So we need to think holistically about reprocessing in terms of patient safety, of course, that's the number one driver, but how the industry is going to react to the challenges we're facing.
Yeah, sure, and I appreciate that, in other countries elsewhere in the world it's a problem, but I mean, we're in Florida right now, and we suffer drought conditions, and then water is rationed. So even here,
California comes to mind.
California comes to mind.
Iran is probably one of the biggest ones I can think of, just because their water table is practically disappeared.
Yeah. Interesting. So maybe you could just update on. I think you were sharing with me. You're very long in this latest project, and just maybe you could just update us the status and where you. And then any expectations of where it might go or next steps after this?
I think first we need to keep exploring this, and the exploration portion, the experimental portion is challenging because of the equipment we need for that, right? So sterilizers are designed to prevent superheated steam, right? We are not designed to prevent things to happen as human, right? It's the human condition to modify things and try to solve problems. But the main situation that I have experienced is that people tweak the performance of the sterilizers to prevent wet loads.
Wet loads,
right?
okay.
And that eventually drives the temperature dries the steam and eventually drives the presence of superheated steam. So I think first we need to continue exploring this. But back to the result, it seems that there seems to be a correlation between the exposure to superheated steam and the reduction of the ability of the sterile barrier system, peel pouches in this case, to maintain sterility. So there is a trend there, and we need to acknowledge that. Whether eventually we may have to change the testing standard for SBS. Maybe we don't have to and just have to focus on educating the reprocessing community, the SPD folks, that this, even though it's helping you to deliver dried loads, is not the right practice. And the detrimental effect is going to be observed on the sterile barrier systems.
No, that's fascinating. I happen to be wearing the KIP, which is which started out with Kilmer, and it's the packaging Kilmer collaboration group. And one of the things, you alluded to is talking about looking at things in different ways. There's been a great collaboration between packaging engineers and industry, and then folks that work at healthcare organizations and responsible for device reprocessing. And one of the things is, we learned very early on is a lot of times we use the same terms, but they don't mean the same things.
Yep.
And then the other thing is that packaging it, when we're validating, we're supposed to do worst case, right? But a lot of times, as packaging folks, we don't really know what's happening inside the healthcare facility and some of the things you're describing. And conversely, with Share, the healthcare folks also don't really completely know what's been done and what's been validated and what they need to do to follow the IFU and to comply. So what you're alluding to is we've discovered in our group and that group, that collaboration group. So next steps. I think you mentioned that you're going to be presenting at the AAMI Exchange, which is in June in New Orleans. And
That's scheduled, tickets booked, and hotel as well. So the expectation is to disseminate that knowledge, and that's going to follow a paper that I'm going to be submitting. So hopefully that's going to get published. So we can, if we publish, we need to help disseminate that knowledge, right? And I think that's going to help. Like kind of my personal mission. I feel very connected with one thing, which is bridging how we manage things from the industry and connecting that with healthcare practices, which is two different animals, and we need to bridge the efforts from the development of IFU for medical device reusable medical devices, but also to help the folks on the SPD navigate the complexity of the IFUs and how they can better streamline the processes. Every single hospital is always running out of time on the reprocessing because there's not enough instruments, right? So how we as the industry, we can help them navigate that and streamline their processes, their throughput, so they can deliver medical devices when they need to be ready on the OR.
And ultimately the goal of all of ours is patient safety and delivering the proper care, good care, safe care to the patient. And so these efforts, the effort that you're undertaking is very much in line with that. And so we appreciate your efforts.
My pleasure.
And maybe if you don't mind just taking 30 seconds, you started out, which was great, but one of the reasons, we're delighted that you're here. And he walked up to me at the foundation booth. I'm like, oh, we wanted to get an interview with you. And immediately, so Dr. Leiva immediately said, yes, of course, I'd be happy to do that. So, but maybe just as you said at the beginning, and maybe just share with other researchers out there the opportunities that the AAMI Foundation offers.
I mean, I think the number one challenge we all face is resources, right? So we're going to put money out of our pockets or not, how we're going to manage that. And when we think about high-end research, say DOE or NASA or NIH, it's extremely difficult to get funding from those sources. However, if you do, you're going to get several $100s to do your research. But that's mostly focused to university and proper research organizations, right? So I think the beauty of the Kilmer grants, both of them, is that people that are not connected to academia, like myself, we can still have access to resources to do that research. When I think about like the what I spend the money on, it was mostly testing, mostly enabling access to testing facilities, but also sending things to a third-party lab, because I don't have, even though I would love to, I don't have testing equipment in the basement of my house, right? So I need to send that to an ISO accredited lab so they can properly test that and provide the information. So the evidence that I'm building is going to be able to withstand the scrutiny. So I would definitely encourage everyone, and there's a broad community across AAMI, but also PDA now and the Kilmer Conference, that of people that may be benefiting from applying to these resources. It's a very streamlined process. I have applied to different funding opportunities, and this is very streamlined. You have to complete a form, you have to provide your story and argument, everything, but it doesn't require a whole lot of work for people to submit that. And I mean, what if I get the resources? What if I do help the industry to advance one tiny step forward in terms of understanding the challenges we're facing? So I invite everyone.
That's greatly stated.
Where are we going to find this information about this?
So if you go to aami.org /AAMI AAMI Foundation, or just go to AAMI.org, and across the top you'll see AAMI Foundation. Click on it, and then again across the top you'll see information about the foundation, but you'll also see, it'll say information about grants, and then we actually have scholarships. You alluded to that as well, and we do try to make it as easy as possible. And the other thing, the foundation, we're funding a broad section of study. So in a way, it makes sense to have it be wide open. But I appreciate you saying that. The only thing I have to say, because I know I'm going to get to see in New Orleans in June, we won't have to be worried about dry heat.
No, we will not. We will not for sure.
Low-hanging fruit, I love it. Well, it was great hearing this story. I love these discovery-based science research stories, and thank you so much for sharing it. Ralph, thank you for pulling him in. This is always a good conversation, and enjoy the rest of the conference, guys.
Thank you.
Yeah.